So today we'll be going over some stuff about genetics. This will be continued in the next science video where we'll go more in depth into the other subjects. So in genetics, there are different components to it. There's the nucleus, chromosome, gene, and DNA. Many scientists questioned what was responsible for transferring genetic material. We already knew that chromosomes were made up of protein and DNA. What Was it the protein or the DNA that transferred the genetic material? What is genetics? It's the study of how genes are passed from generation to generation. The study of how genes give an organism its traits, and a gene is segments or parts of a chromosome. Chromosomes contain DNA and proteins, and have genetic information that is passed down to family members. And DNA is the instructions for each trait. The three functions of a gene: controls the making of proteins, called protein synthesis, directs the order of amino acids, and determines the traits and function of the cell. Name of some traits: hair color, eye color, height, and weight. A cell has 23 pairs of chromosomes with a total of 46 chromosomes. Chromosome is made up of genes, and the genes consist of DNA, which is made up of 4 chemical letters. The two types of nucleic acids: deoxyribonucleic acid, which is found in the nucleus, and ribonucleic acid, which is found in the cytoplasm. The difference between DNA and RNA. DNA is double-stranded helix, deoxyribose sugar, nitrogen bases, which we saw before, which include A, T, C, G, which are the chemical bonds. One type of DNA and connected by weak bonds. RNA is single-stranded, ribose sugar. The nitrogen bases are A, U, C, G, so they're both the same besides the second base, which is T and U, and are 3 different RNA types. DNA acts as a template, meaning it acts as a pattern so it can be copied. DNA is copied before mitotic and meiotic cell division. 3 types of RNA. There's mRNA, messenger RNA, which copies DNA, carries the instructions for protein assembly. tRNA, which stands for transfer RNA, and transfers or carries amino acids. And rRNA, which is ribosomal RNA, which makes ribosomes and cytoplasm site for protein synthesis. DNA replication, complementary strand. DNA replication, which is the copying of DNA to have a complete set of DNA molecules. Over here are the chemical bonds, and the ones they pair to. So A is paired with T, and G is paired with C. Here's how DNA replication works. So here's the parent molecule, which is the original strand of DNA. And as you can see, we can see the bonds here from A to C, A to T, C to G, T to A, AT, and GC. The first step in replication is to split the two halves, or the bonds. Then, each parental strand now serves as a template for new strands to connect. Then, once it's all connected, then now we have 2 copies of the original strand. Every protein has a specific sequence, shape, and function, and all these determine their job and what they do. What does a gene do? Sequences the order of amino acids for a specific protein. A change in the sequence of a base will change how mRNA will transcribe for a specific protein. Different protein could be made. What does gene expression mean? It means a gene is on, so it's making protein. When gene is off, the gene is not being expressed and no protein is made. What will cause this rabbit's color to change? Gene expression and the environment. Changes in temperature causes change in gene expression. Cold temperature turns on the gene that controls the black pigment. Gene mutations. What is a mutation? A permanent change in the structure or amount of genetic material. There are two causes for mutations. Natural random errors happens during DNA replication with DNA polymers. And mutagenic. Chemicals and radiation damage DNA. Examples such as X-rays, UV light, radiation, and toxic chemicals. Some examples include the albino koala, the albino peacock, and albino alligator. A very famous example is Snowflake, the world's only captive albino gorilla. Died in 2003 from skin cancer, lived past 40 years old at the Barcelona Zoo in Spain. Albino, which means no skin pigmentation. Both parents carry the recessive gene for albinism. However, the parents themselves were of normal color. Some mutations are helpful, neutral, or harmful. Genetic variations result in new traits, which could result in better working proteins. Neutral is a change which equals to an unchanged protein, so the function stays the same. And defective proteins, which means non-functional proteins, which can be harmful to the organism. Two types of mutations: gene mutations and chromosome mutations. A change in one or more bases within DNA. It affects a gene on a chromosome. Change in the number or structure of chromosome. Gene mutations. Alter amino acid sequence, alter protein shape, and alter protein function. Chromosome mutation types. There is translocation, which two different chromosomes change parts with each other. There's inversion, which is flip genes. Addition and duplication, which is added genes, and deletion, which is missing genes. And that's all for the genetic unit.